The Krebs cycle, or the tricarboxylic acid cycle, takes place in almost every cell, where it produces energy and reducing power. This cycle is not conceived as tightly regulated, unlike other metabolic processes, such as glycolysis or gluconeogenesis. It is increasingly evident that the alterations in the Krebs cycle are associated with diseases mediated by autophagy, apoptosis, and oxidative stress. It is relevant the association of the neurodegenerative diseases with alterations in the mitochondria, including Krebs cycle malfunction. We have reported details of Krebs cycle dynamics using an in silico model. Our results suggest that when there is a continuous input or output of Krebs cycle intermediates by the so-called anaplerotic reactions, the Krebs cycle needs tight regulatory mechanisms. Otherwise, it will be difficult to maintain homeostasis. For instance, a continuous accumulation of carbons inside mitochondria acting as cycle intermediates will not increase much the energy production, while it will produce an unsurmountable increase in the concentration of metabolites that would lead to mitophagy. Also, when modeling the cycle in different conditions and looking for the achievement or not of a steady state, the results suggest that the cycle may have a regulation not yet discovered to go from an open cycle to a closed one. Said regulation could allow for reaching the steady state, thus avoiding the unwanted effects derived from an aberrant accumulation of metabolites in the mitochondria. That's why we explored which kinds of regulations would lead to these four scenarios. 1. Control of acetyl-CoA production, which is the main limiting factor of Krebs cycle flux, and ATP production. 2. Control of activity of the tricarboxylic acid cycle enzymes. 3. Control the activity of enzymes related to anaplerotic reactions. And 4. Control the activity of mitochondrial membrane transporters. The tight regulation of acetyl-CoA production by pyruvate dehydrogenase is already known. This enzyme is a multi-enzymatic complex that is regulated by, among others, serine and threonine phosphorylation by kinases that are present in mitochondria. We then opt to focus on the mechanisms on regulation of the activity of Krebs cycle enzymes, especially of the alpha-ketoglutarate dehydrogenase, a macromolecular complex that is structurally similar to pyruvate dehydrogenase. Based on evolutionary evidence, we hypothesize that Krebs cycle could be regulated in an hormonal-like fashion, whose main characteristic is producing a fast and a huge change in enzymatic functions. Hormonal-like fashion means that the activation of receptors in the own mitochondria membrane elicits signal transduction and leads to changes in the level of second messengers, cyclic AMP or calcium, but inside mitochondria. Mitochondrial signaling would lead to post-translational modifications such as phosphorylation of Krebs cycle enzymes. The chances that some of these enzymes might be phosphorylated are high. In fact, residues susceptible of phosphorylation are highly conserved. More details are available in the manuscript. These receptors are likely of the G protein-coupled receptor family, or in other words, it is likely that some of the 100 GPCRs are also expressed in mitochondrial membranes. GPCRs is the most populated family on the mammalian proteome. Their gene sequence is highly conserved along evolution. Interestingly, in recent years, serotonin, cannabinoid, angiotensin, and purinergic receptors have been found in mitochondrial membranes. As one would expect, the type of proteins that participate in signal transduction of GPCRs expressed in the plasma membrane are found also in the mitochondria. Examples are protein kinase A and protein kinase C. Finally, a relevant transcription factor, Krebs, that is phosphorylated upon cell surface GPCR activation has also been found in the mitochondrial matrix. All these findings constitute evidence of hormonal-like mechanisms in the mitochondria. Receptors are there, and the signal transduction molecules are also there in the mitochondrion. This evidence encourages the validation of this hypothesis by experimental methods using isolated mitochondrion and the new molecular methods that did not exist decades ago when the Krebs cycle was a scientific trend. One more piece of evidence supporting this hypothesis is that GPCRs have been identified already for two key Krebs cycle intermediates, succinate and alpha-ketoglutarate GPCR. With this review, we aim at stimulating research on Krebs cycle regulation, something that has not been a trend in the last 3 decades. Working with isolated mitochondria and trying to detect signaling events in these organelles is technically challenging, but this task is worth it. Uncovering GPCR-mediated signaling to modulate Krebs cycle is of paramount importance to better understand metabolism, but also to better understand the pathophysiological events underlying the diseases that are accompanied by metabolic impairment, oxidative stress, autophagy, mitophagy, and/or cell death.